Hi everyone, welcome back to another lecture in the Immunology series. Now, after learning all the basics of the innate and adaptive immune system, we are finally here able to discuss how immunological memory works in conjunction with vaccines. We will not have a very biotechnical discussion about the vaccines, but more so from an immunological standpoint. So let's get started. Here are the lecture objectives. We will first talk about B cells and T cells involvement in immunological memory. Then we will have a very brief overview of the historical aspect of vaccine discovery and development. Now, for those of you who like medical history, you may find that part interesting. Then we will compare and contrast different types of vaccines with a deeper discussion on the seasonal flu vaccine and an overview of how messenger RNA vaccine stimulates immune responses. Lastly, we will look at how vaccine adjuvants work to promote innate response. Perhaps the first documented effect of immunological memory was recognized as early as 430 BC, when Thucydides recorded that while the plague was raging in Athens, the sick and the dying would have received no attention had it not been for those individuals who had already contracted the disease and recovered and recognized their immune status. In today's world, protective immunity refers to the production of antibodies after being infected by a pathogen. Antibodies continue to circulate in the body for several months and prevent reinfection from the same pathogen. The antibodies, either IgA or IgG, have the ability to block the virus from infecting the host by coating the outer surface of the virus and neutralizing it. They can attach to bacterial pathogen and then bind to the Fc receptor on a phagocyte, which facilitates its uptake and phagocytosis as well. Plasma cells maintain low amounts of antibodies in circulation. However, plasma cells are short-lived, and antibody production to a specific pathogen decreases over a period of a few months until a steady state is reached. Now, this steady state is maintained for life by plasma cells in the bone marrow. These antibodies are a form of immunological memory. After primary infection producing effector B and T cells, memory B and T cells will form, and the purpose of this is to make sure future invasions by the same pathogen will be met with an aggressive immune response, so the patient will have less to no symptoms. During a primary response to an infection, the activated B cells differentiate into plasma cells that secrete IgM antibodies. This early pentameric IgM response is an important first line of defense against infections, such as participating in classical complement pathway activation. However, it's tough for IgM to leave the blood because it's bulky. It has trouble getting to the tissues. Somatic hypermutation and isotype switching lead B cells to stop producing IgM and to produce high-affinity IgG and IgA instead. IgG is the dominant antibody, and IgG is small and can travel to extracellular spaces of the tissues, unlike IgM. When the body is exposed to the same pathogen, memory B cells are rapidly activated and differentiate into plasma cells that secrete large amounts of high-affinity IgG specific to the pathogen. This rapid and robust response is known as the secondary response and is much faster and stronger than the primary response. IgG antibodies have higher affinity and increased effector functions compared to IgM, which makes them more effective in binding to and clearing the pathogen. The IgG blocks pathogen from activating IgM production in naive B cell. Memory T cells are a specialized subset of T lymphocytes that are generated during an initial encounter with a pathogen or vaccine. Effector T cells will outnumber memory T cells during the primary response, but there will be memory T cells synthesized that will recognize the pathogen in future infections. During the secondary response, both memory CD8 and CD4 T cells are activated. Memory T cells can be broadly classified into two subtypes: central memory T cells, or TCM, and effector memory T cells, or TEM. TCM cells circulate in the secondary lymphoid organs and tissues and have the ability to proliferate and differentiate into effector T cells upon interactions with dendritic cells. On the other hand, TEM cells are tissue-resident memory T cells that have migrated to the peripheral tissues during an initial infection or vaccination. They express a low level of integrin, which allow them to quickly migrate to site of infections or inflammation and be activated by antigen-presenting cells at the site. Once a pathogen has been eliminated during primary immune response, the number of memory T cells will outnumber the number of effector T cells for the same pathogen. This is immunological memory. In summary, activated naive T cells and B cells can become effector or memory cells. Effector cytotoxic and helper T cells and plasma cells are short-lived and provide adaptive immunity during an active infection or immediately after vaccination. On the other hand, long-lived memory T cells and B cells would reside in bone marrow, secondary lymphoid tissues, and peripheral tissues and get reactivated quickly upon the secondary encounter with the same pathogen. Now that we have some understanding of how immunological memory works, let's have a look at how it is the basis of vaccination. I understand vaccination is no longer a purely scientific topic since 2021, and the field is rapidly changing with the new mRNA technology. We are here only to focus on more of the classical and well-established knowledge of vaccines. Let's look at the past. To talk about the past of vaccination, we would have to trace back all the way to ancient Egypt and look at smallpox. Smallpox has an ancient lineage. Egyptian writing 5,700 years old described this disease, and there is a mummified pharaoh in the Cairo Museum who died in 1157 BC that has pustules indicative of smallpox on its face and hands. Ramesses V reigned for only 5 years during the 20th dynasty. He died in his early 30s, and a possible reason for his premature death is evident on his mummy, which is scarred on the face, neck, and chest by smallpox. Smallpox is caused by a virus, the Variola virus, which has been part of the human population for thousands of years. The Variola virus has been eradicated now from the human race, but still exists in stockpile in the laboratories for the purpose of research and for developing vaccines. The smallpox is spread through face-to-face or touch-to-touch contact between human beings or through contact with infected bodily fluids, such as the pus that seeps from smallpox sores. It can also be spread through contact with things that are infected from a person. and has been contacted with, such as sheets or clothing. People become contagious when their fevers start, but they are at their most dangerously contagious once the rash starts. It kills about a third of its victims and leaves the rest scarred, blind, deaf, or crippled. There is no specific treatment available. However, survivors are immune to future infections. And the early form of a vaccine comes in the form of inoculation or variolation, was designed to target smallpox. Now, Lady Mary Woodley Montagu was an English lady who was struck with smallpox in 1715. She recovered but was deeply pockmarked. Now, her younger brother died of smallpox. As the wife of the British ambassador to the Ottoman Empire, she traveled with him to the east. There she discovered that an ancient practice of inoculation was practiced. In Turkey, she witnessed practice of inoculation by exposing a healthy person to infected material extracted from lesions of a person with a mild or minor smallpox. It turned out that intranasal or intradermal application of a live virus generally produced a mild disease that provided immunity from further infection. However, there was a fairly high risk. In approximately 1 in 100 people, variolation produced a fatal case of smallpox. And in 1718, Lady Montagu had her young son, Edward Jr., inoculated. In 1721, back in England, she had her 3-year-old daughter inoculated by the doctor, Dr. Charles Maitland, during a smallpox outbreak. Therefore, the daughter became the first person known to be professionally inoculated in England. Now, testing was done on prisoners in Newgate Prison in London. Now, prisoners were inoculated and then exposed to people ill with smallpox. Now, those were the research in the old days. This is medically unethical by today's standard, but back in the 18th century, it was okay. After this clinical trial, quote-unquote, seeing this method was relatively safe and effective, the royal family then began using it. And back to the U.S. soil, looking at U.S. history, perhaps the most decisive decision George Washington made was to inoculate all the soldiers to prevent smallpox. During the 9-month siege of Boston in 1775 and 1776, at the time the city was reeling from the epidemic. Approximately 10,000 American troops marched on Canada in fall of 1775, and at one point, nearly 3,000 of them were sick. And by the spring of 1776, the conditions of the American soldiers in Canada had deteriorated severely due to continuous outbreak of smallpox. Approximately half of the soldiers were ill. Fully aware of the disaster in the North, George Washington realized that merely evading smallpox would no longer suffice. He wanted to prevent it altogether. Looking at U.S. history, perhaps the most decisive decision George Washington made was to inoculate their soldier. At first, Washington simply required new recruits to be inoculated. Then in February 1777, he bit the bullet entirely to require everyone in the troop to be inoculated. This was a bold move. At the time, variolation was technically outlawed by the Continental Congress. So Washington was openly floating the law. Whole divisions were inoculated, and those who were sick were quarantined, a process that would continue for months. At the same time, strict secrecy was maintained to prevent the British from uncovering the program, so that they could not launch an attack upon the recovering troops. By the end of 1777, 40,000 soldiers were immunized. The result was stunning. The smallpox infection rate in the Continental Army rapidly fell from 17% to only 1%, prompting the Continental Congress to legalize Variolation across the states. Fast forward about a decade after the George Washington's order of inoculation, Edward Jenner, who developed and propounded vaccination in the 1790s, is often credited as the founding father of immunology. English physician Edward Jenner noticed that milkmaids who developed a less serious cowpox disease did not develop the deadly smallpox. In 1796, Jenner took the fluid from a cowpox pustule on a dairymaid's hand and inoculated an 8-year-old boy. 6 weeks later, he exposed the boy to smallpox, and the boy did not develop any symptoms. Jenner coined the term vaccine from the word vicar, which means cow in Latin. Immunity to one virus conferred immunity to a second as well. Vaccine success was based on an antigenic determinants shared between Vaccinia and Variola virus. The vaccination with cowpox virus elicited neutralizing antibodies that react with antigenic determinants shared with smallpox virus. Shared antigenic determinants of cowpox also elicit protective T-cell immunity against smallpox. However, vaccination using live virus is usually not safe. Most pathogenic viruses also do not have a safe counterpart, and vaccine production also problematic until well into the 19th century. That was when microorganisms were first isolated, grown in culture, and shown to cause disease. Nevertheless, vaccination with live Vaccinia cowpox virus was much safer than inoculating with smallpox virus. The widespread of practice eliminated smallpox in the 1970s, but there are some luck involved. First, we were lucky to have a cowpox virus that was similar enough to smallpox, and it was not deadly. Second, smallpox is only found to infect humans. If the virus jumps between species, then it would not have been so easy to eliminate it. Nowadays, different types of vaccines are utilized to prevent the spread of pathogens. We have live vaccines. These days, we use mild pathogen related to harmful pathogen and is injected to skin. Examples, like we talked about, the cowpox pustules. We also have live attenuated vaccine. These are weakened pathogens, usually done by chemicals, heat, or radiations. They generally grow very poorly in human cells, and they grow in animal cells. The examples of live attenuated vaccines are poliovirus vaccine and MMRV vaccines. And we also have killed vaccines or inactivated whole virus vaccines. They are fully attenuated microorganisms, and their genome has been completely destroyed by irradiations or chemicals, so they do not have any ability to replicate in any living cells. Examples are influenza virus vaccine and also some poliovirus vaccine. In addition to those, we also have recombinant vaccines. Recombinant vaccines utilize recombinant technology, where the gene from a specific pathogen is cloned or incorporated in another species to produce proteins that are immunogenic. And these surface protein productions are usually in large quantity and at lower cost. Examples of recombinant vaccines are human papillomavirus (HPV) vaccine and hepatitis B vaccines. We also have conjugate vaccines, where proteins are conjugated to polysaccharide surface antigens derived from pathogen. Remember, polysaccharides are not immunogenic from T cells' standpoint. They do not react to polysaccharides, so they need a protein conjugate to link to the polysaccharides to stimulate T cell response. And therefore, in this case, it is both able to be recognized by both B cells and T cells. And examples of conjugate vaccines are meningitis vaccine. Then we have toxoid vaccines. Now, toxoid vaccines are as a modified version of a toxin that is secreted by usually a bacteria, and vaccine against secreted toxins and rather than against the specific pathogen. Examples are tetanus, diphtheria, and pertussis. Then we have newer versions of the vaccine, messenger RNA vaccine and viral vector vaccines. They are different, but they do share some similarity, where they deliver a genetic blueprint of an antigen to host cells and relied on the host cells to synthesize the antigen that trigger the immune response. Regardless of what type of vaccine, a successful vaccine requires initiating pathogen-specific immune responses. The vaccine's effect will be dependent on the infectious agent. For extracellular organisms, CD4 T cell will dominate to stimulate more humoral responses against bacteria. For intracellular organisms, CD8 T cells are required to kill virally infected cells directly, and humoral responses are needed to neutralize free viruses from the blood and tissues. Site-specific vaccination, such as from a mucosal surface vaccine, is particularly helpful in providing host defense at the point of entry. In this case, plasma cells will secrete both IgG in blood and IgA at mucosal surfaces, such as the nasal cavity of the respiratory tract. Now let's have a deeper discussion on the seasonal flu vaccine. Most of the seasonal flu vaccines are killed vaccine or inactivated vaccine containing 4 different influenza strains against seasonal influenza virus. These vaccines are produced in egg, chicken egg, or cell culture. Now these viruses are grown in these eggs and cells and is purified and killed at the end. Now the vaccine can stimulate immune response, innate immune response first, by allowing recognitions of molecular pattern expressed by the virus, such as the HA and NA glycoprotein. Now, and then antigen presenting cells is going to digest and express flu antigens to stimulate both humoral and CD8 T cell responses. For people who are allergic to eggs, they would need egg-free flu vaccines. Now, these vaccines are grown in animal cells. Now, there are also alternatives, such as recombinant flu vaccines. Now, the recombinant technology incorporates genes for the HA glycoprotein and incorporate into an insect virus, baculovirus, and utilize that virus to transfect insect cells in culture and to produce a large amount of recombinant hemagglutinin, and afterward these proteins are going to be purified and packaged in a vaccine. Now in this case, these recombinant HA protein will require adjuvant to be added to increase the innate response. The innate immune system is the body's first line of defense against invading pathogens. Toll-like receptors recognize a wide range of conserved structures on microorganisms called pathogen-associated molecular patterns or PAMPs in live attenuated, and inactivated vaccine, but they have difficulty binding to purified polysaccharides of conjugated vaccines, so the innate response is decreased. Vaccine adjuvants are substances that are added to vaccines to enhance their effectiveness by boosting the body's immune response to the vaccine. Adjuvants work by stimulating the innate immune system, which in turn enhances the adaptive immune response to the vaccine antigen. There are several examples of vaccine adjuvants. For example, aluminum salt, also known as alum, are the most commonly used adjuvant in human vaccines. Alum works as an adjuvant by forming a depot at the injection site that slowly releases the vaccine antigen. This prolonged exposure in turn leads to activation of more antigen-specific T and B cells and the production of more antibodies. Oil-in-water emulsions are a type of vaccine adjuvant that work by enhancing the delivery and presentation of the vaccine antigen to the immune system. These adjuvants consist of an oil-based phase and a water-based phase that are emulsified together to form a stable suspension. When the emulsion is injected, the oil droplets in the emulsion are taken up by immune cells such as dendritic cells and macrophages at the injection site. The oil droplets are slowly metabolized by the immune cells, which results in the prolonged release of the vaccine antigen. This sustained release of antigen promotes the activation of antigen-specific T cells and the production of antibodies. The oil droplet also serves as a depot, which helps to localize the antigen at the injection site and enhance the immune response. Third, we have toll-like receptor agonists such as CpG oligonucleotides. It works by activating the innate immune system through stimulation of toll-like receptor 9, a receptor expressed on immune cells such as dendritic cells and B cells. When CpG oligonucleotides are included as an adjuvant in a vaccine, they are taken up by the immune cells at the injection site. Once inside the immune cell, CpG oligonucleotides are recognized by toll-like receptor 9, which triggers the activation of a signaling pathway that leads to the production of pro-inflammatory cytokines and the upregulation of co-stimulatory molecules on the surface of the immune cell. Now let's look at other notable examples of vaccine that are designed against bacterial toxins. Toxins secreted by bacteria can lead to disease and death. For example, tetanus is a disease caused by secreted toxins from a soil bacteria, Clostridium tetani. The bacteria can enter the body through breaks in the skin. Their spores affect motor neurons through the production of neurotoxins, tectinospasmin. The toxin blocks neurotransmitters and can lead to severe muscle stiffness and spasms, and even death. Fortunately, vaccination with the tetanus toxoid vaccine can prevent infection. Bacterial toxins can be converted into toxoid that can promote immunological memory. Technospasmin is chemically modified into a non-toxic toxoid protein. The toxoid is injected to promote immune response and stimulate immunological memory formation in adaptive immunity. The tetanus vaccine is given in combination with other toxoids in a combo vaccine containing diphtheria, pertussis, and tetanus toxoids. There are many versions of this combo vaccine and is given according to different age groups. Now let's wrap up this lecture with a very brief talk on messenger RNA technology, which is a term that most people have heard and experienced since 2020. Now I know this is a rapidly evolving field, and here I'm only presenting a snapshot of the established knowledge from a technical standpoint. The idea of using messenger RNA as therapeutics is not as new as a lot of people believe. Now, in the early 1990s, researchers discovered that messenger RNA could be used to produce proteins in cells outside of the body. However, it was challenging to get the messenger RNA to enter cells inside the body where it could be used to produce proteins. Now, in the early 2000s, scientists began to explore ways to overcome this problem. They discovered that by modifying the messenger RNA molecule, they could increase its stability and enable it to enter cells more easily. They also found that by incorporating nucleoside modifications, they could reduce the immune response that was often triggered by messenger RNA. Now, simultaneously, different teams of scientists were looking for ways to deliver messenger RNA into cells. Among all of the tested RNA delivery platforms, lipid nanoparticles are the most versatile. Now, lipid nanoparticles are small spherical particles composed of a lipid bilayer that surrounds and protects the messenger RNA molecule. The lipid bilayer is made up of a mixture of phospholipids, cholesterols, and aminolipids, which are chosen based on their ability to form stable and biocompatible nanoparticles. The messenger RNA is encapsulated inside the lipid nanoparticle, which protects it from degradation and helps it to enter cells. The lipid nanoparticle can bind to cell membranes and fuse with them, allowing the messenger RNA to enter the cytoplasm of the cell, where it can be translated into the desired protein. After the messenger RNA enters the cytoplasm of the cell and gets translated into proteins, the fate of that protein will depend on its origin. If the protein is native and recognized as a self-protein, it can perform its function as intended. However, if the translated protein is non-self, such as from pathogens and cancers, it will get broken down into peptide pieces and presented via the MHC class I molecule. Now, subsequently initiate adaptive immune responses with the mechanism we talked about in the Immunology series lecture 6. So if you need a refresh on that concept, please check that out. To conclude this lecture, in summary, immunological memory is the basis for vaccines. And a successful vaccine needs to be safe, protective, and induce long-term immunity. The field of vaccine is rapidly evolving with new technologies, and we need to be aware that no vaccines are risk-free, and risks and benefits must be weighed carefully for each vaccine and for each age group. That is everything for this lecture, and I will see you in the next one. Take good care. Bye.